we'll get things rolling here. I'm Chris McMaster. I'm the chair of the Department of Pharmacology, and I'm the lead investigator on the IGNITE project, which is one of the sponsors of this series. And it's my pleasure to introduce Kym Boycott. Kym did both her MD and her PhD at the University of Calgary. And then from there she moved to CHEO, the Children's Hospital of Eastern Ontario, where she's a neurogeneticist. She has a Faculty of Medicine Research Chair in Neurogenetics, and she's also a CIHR clinical investigator. She's won the Sick Kids Young Investigator Award, the March of Dimes Young Investigator Award, and she's also the lead investigator on probably the biggest, baddest gene discovery project on the planet right now called FORD. And Kym's here to tell us exactly how successful that thing's been and why and where they're going next. So take it away, Kym.
Well, thank you, everyone. I'm very pleased to be here. I didn't know why. Sarah sort of invited me, and I didn't know it was such a big thing. And there would be lunch. So I'm thrilled. I'll have my lunch after. And I'm going to talk to you today about our FORGE project, which is essentially a nationwide effort to identify genes for rare disorders. And we've been going now for a year and a half. So I want to summarize sort of at a very high level what we've done. And something that really has become, that initially I just ignored, but has really become a key thing to recognize is that Canada has a different sociology than a lot of other countries, and that we're successful because of this. And lastly, to talk about how rare diseases is an example of, and a great example of personalized medicine. So I'm not going to talk a lot of background because Chris and others tell me that most of you will know quite a bit about rare diseases and already appreciate why it's important to study them. But if you look at that slide over there, essentially it's predicted we have about 7,000 rare diseases. We understand the genetic etiology for about half of those, that's the dark green. But then there's probably about 3,500 or so where we suspect that it's a rare disease and that there should be a single gene cause for it, and we don't yet know it. Some of these have been reported as phenotypes in the literature, and so they're recognized as a group of signs or symptoms together. Others are things we see in our clinic, and we just really think that it's genetic, but we haven't described it or published it. To put that in perspective, there are currently in Canada genetic testing available for only 150 of the 3,500 disease genes. And as I've talked to many of your colleagues today, that's a big issue. Especially in a place like the Maritimes too, where your out-of-country budget, as it is for most of us across the country, is limited. So we can't necessarily get access for our patients to get these diagnoses. And then to put it in perspective again, and something I think that you probably all understand because of IGNITE, is that there's definitive therapy for only 200 of these. So there's a great medical need. And so to skip right along to how this all happened, in July, of 2010, we got together because there was an opportunity put in front of us that if we established a national consortium of the genetic centers across the country, we might be able to do something really big in rare diseases. And so with about 2 weeks of notice, we contacted all of the clinical centers, genetic centers across the country. And even though it was the summer, we were able to find a representative from 16 of the 21 centers to come on 2 weeks' notice in the middle of the summer. We collected in Toronto, and Sarah was there from Halifax. We established the consortium, and we had a representative from each of the centers who was engaged in helping move this along. But essentially all members of the Canadian College of Medical Genetics are part of FORGE, as well as other specialties like ophthalmology, neurology, cardiology, any physician who sees patients with rare disorders. And we also have about 50 scientists. These include basic scientists, informaticians that are all part of this group for about a total of 150. And we currently, it also includes some, hopefully an emerging population of genetic counselors, and also laboratory directors, which I think is another group we need to target a little bit better. And we're currently collaborating with 17 different countries. And so what we put together in that sort of 6 months up to funding was our two priorities, which was the way that the funding opportunity was described is we had to rapidly, and they meant rapidly, identify genes for rare disorders. Initially, the project was going to be 1 year. This was 12 months. This was a directed grant. You better move. You better be ready. And I've done 6 months' work prior to. It's now a 2-year project, and we've received several directed grants after this to make it so it can last longer. And the second thing, though, that we really all felt as the clinical community is that we needed something more sustainable. Just got to be more than 2 years for rare diseases. We have to have something that can continue from this, and I'll talk about that at the end. So it began in April of 2011. We're about 16 months in, I guess, and we will finish in March of 2013. And so right now we're trying to determine what we're going to do next. So essentially, I just want to talk a little bit about how it works before I will describe sort of the overarching things we've learned from this. So we've had over 375 diseases proposed to us for study. They've come from everywhere in the country. And we have a look at them and sort of try to decide those that are most likely, because of the way this funding is put together, to result in a novel gene discovery. They have to meet a certain number of criteria, which are there in green, but our focus is on pediatric diseases. We don't study things that are adult onset. That was just part of the initiative. There has to be at least one Canadian patient for which it will be relevant. So we don't study things from the Middle East for which we have no Canadian patients for which it will be relevant. It has to come from Canada. It has to likely be caused by a single gene, and it has to, the families and patients have to have had a fairly good workup. But we really recognize across the country that not every province is going to have the same access to testing, and so we can't sort of prefer patients from one province because they've got better out-of-country funding than another province. So we're very cognizant that workup had to be best for that particular province, and that sometimes we're going to discover things that were already known. And so essentially it looks like this in terms of how many disorders were put forward to us and that we will study from across the country. There's representation from almost every province. No families from Yukon or Northwest Territories that I know of, but there is a couple of disorders being studied from Iqaluit, though they come in via Quebec. And essentially what the approach is, and I won't go into a lot of detail about this part, but we use exome sequencing primarily. And it's done at 3 of the Science and Technology Innovation Centers, one in BC, one in Montreal, and one in Toronto. The one in Montreal is the one that the IGNITE project uses as well. And what happens is when the disorder is going forward from, and it's been decided to move forward and all the DNA is ready, it may be coming from multiple places in the country. So it collects at the particular innovation center, and they're generally run together so that they can be analyzed together and we get the answer as fast as possible. And what happens when this data comes back is obviously we are dealing with a very high volume. In Montreal, we run 96 samples at a time. And so it goes back to the project team, which is the clinician and the laboratory scientists, the basic scientists who initially proposed the disorder. If a clinician has this disorder they would really like to study, but is not linked up with a lab that can validate, we make those arrangements for them. But the data primarily goes back via our informatician who's done their analysis to the project team, and the project team decides the next steps and keeps us up to date as to how it's going. Our data also collects at our National Data Coordination Center, so all of the foraged data is being collected at the University of Toronto, and we do stuff to it, kind of like that. So we make sure that the QA looks good, the QC looks good, that the SNP arrays actually match the exome data, that we haven't screwed up any patient samples, the quality is equal across the 3 different innovation centers. And then we also collect these variant data sets for specific populations in Canada, like French Canadians, Hutterites, Mennonites. And then also from that, we have recently developed a phenotyping tool that will be used with the variants to share internationally for those disorders we can't solve. The other thing Forge is doing, and in part with Conrad Fernandez, who is part of the Cancer Consortium, and also part of IGNITE, is to try to work with, we're working with Bartha Knoppers, and what they're trying to do is develop Canadian guidelines for return of results to pediatric participants that are incidental in nature. So I suspect this is something many of you have heard of lots before, but the question is, what about the other, so we usually get 500 variants from an exome, what about the 499 or 498 that aren't causing the disease? What do we do with those? And we have looked at our data, and you tend to find 0 to 1 significant finding per exome. And it's the question of what do you do with that when it's a breast cancer mutation in a child? And how do you return that? And so we're working on actually developing those guidelines. So I want to go through the ways that we have approached novel gene identification and just give quick examples to illustrate. So the first and probably most powerful category is to look at patients or families who are unrelated to each other who have the same rare disease. So the example here is a big long name, mandibular facial dysostosis with microcephaly. Essentially microcephaly, small head, and mandibular facial dysostosis is underdeveloped mandible and usually cleft palate. So the first baby, A there, is a baby that was born at CHIO in 2005, for which had this unrecognizable syndrome. The syndrome wasn't described until 2006. She was one of my first patients, my first month on call. And after this first description was described from a group in Brazil, and then later, we sort of identified the 10, 12 patients in the world that had this, so only one from Canada and the rest from somewhere else. And we put them through the exome. And so what that looked like is we sequenced, in the end, 5 individuals who we thought had the classic features. If you look at all their variants together, they have 2,500. If you look at those that are rare, there's 1,500. And then you start to look sharing between 2 patients. So every time you add a patient, you decrease the number of variants. So 60 to 8, and then to 4. And then when you looked at those 4, there are some things like false positives in the exome. You could get rid of the other 3. And the gene was EFTUD2. The 5th patient ended up having a deletion of this gene. And so we were pretty happy that we had the disease gene there. And that one's been published earlier this year in the American Journal of Human Genetics. The second approach we use, which is something that I think that IGNITE also uses, is we use families that we can use mapping information to decrease the search space. So these are either consanguineous families or dominant families with enough affected individuals that you can get rid of part of your genome. And so essentially the example that I'm showing here is a family with 4 individuals with intractable epilepsy and microcephaly. And we mapped this to a region on chromosome 19. We just did the exome of 1 person. And in that, when you look through that exome and just look then focusing in the homozygous region, there are 3 variants. And one of them is in a gene that was reported 2 months earlier than us finding this, that causes this particular disorder, which is WDR62. And so this is actually a pretty efficient way, because as clinicians, you'll know that the workup of microcephaly and intractable seizures could be very expensive if one went to town on it. Probably cost you $25,000 of out-of-country testing. And this is basically 4 SNP arrays in 1 exome. Probably we got it for about $4,000. The other approach that we took, and initially when we discussed this, when Sarah will remember when we were sort of sitting around, how are we going to do this? This was a category we weren't sure we're going to be very successful at, but this is very common. 2 SIBs, no other family history, they both have it. So we did go through, and this has actually been a very successful category for gene identification. And so this is 2 brothers who were previously well. They developed hearing loss in early childhood, later cerebellar ataxia and neuropathy in their legs, and then most recently the oldest has retinitis pigmentosa. So this is a degenerative condition of unknown etiology. So we exome sequenced both the boys very early on in forage, which is why our variants are higher. And essentially they only shared 2 genes, and one of which we could prove biochemically was the cause. And so they have a novel and new type of D-bifunctional protein deficiency. This is a metabolic degenerative disorder, but they are absolutely, it's a new type. So they're initially well, and then develop this degenerative course over time, meaning that they have residual function of their enzyme, but over time they accumulate damage. And then what we would normally do clinically for screening for this type of metabolic defect is all normal in them except for the very, very specific enzyme testing. So our normal screens would not have picked this up. The other 2 categories we look at is these are the ones, category 4 is patients who might all have a phenotype like microphthalmia or small eyes. And you'd sequence maybe 20 of them and see who don't have a cause identified and see if you can find something common between them. Category 5 I'll talk to you about at the end, which is 1 patient, no family history, no consanguinity, but suspect they have a genetic disorder. If you just sequence them, what happens? And the 6, which I want to show you one example of because I think it's really interesting, it shows the power of this approach, is looking for somatic mosaicism. So not necessarily germline mutations, but things that happen after birth, they still cause rare disease. And I can explain that a little better, but the power of this comes from sequencing different tissues from the same patient. So often we go back to pathology samples, or we use skin fibroblasts, even saliva or renal through the kidney is a good way to do it. And so to show this, This is a disease called megalencephaly capillary malformation syndrome. It's characterized by overgrowth, so the kids become very large after birth. Often by a year of age, they have the head the size of an adult male, and it continues to grow, so their head becomes very big, and that's shown there with the MRI. Brain becomes very large. The brain pushes down and causes problems such that the brain needs to be shaved off regularly because it's causing compression of the spinal cord. They have often developmental defects in the way that their brain forms, so there's little bumps instead of having sort of more round bumps, which causes seizures. They can have those finger abnormalities there, so syndactyles, fused fingers, and polydactyles, extra fingers or toes. And they have this sort of doughy skin, and they're quite flexible, which is hypermobility. But when you look at that group, there was a second group where we weren't sure if these were all one thing or two different things. Turns out this is more confusing than all that. But we sort of selected a group that had an extra finger versus not, and then we analyzed them all separately. And so what we ended up doing, this was the first set of exomes, 8 exomes we put to this project that we did as part of Forge. It was one that was led out of Ottawa. It was one of my team projects. And we were so depressed because we couldn't find it. So we'd done 8 exomes. This is our very first project. This is Forge 1. Nothing. So back then, we were sort of thinking, this is not going to go very well. This is a very classic disorder. This is very recognizable. Why can't we find it? So we ended up teaming up with, so if you can't figure out yourself, you should collaborate. So we ended up teaming up with Bill Dobbins in Seattle, and they had sequenced some families because they had familial recurrence. So they had one family where both parents were fine, but they had 3 kids who were affected. And those 3 kids gave us the biggest clue in the sense that they had a germline mutation and gave us an idea of what pathway we should then go looking at. And so what happened is all of our patients have the first one, which is MCAP, and they all have mosaic mutations in this PIK3A gene, which means that their mutation is not 100%. So if we're looking at, if you don't know where to look, it's really hard to do this systematically. But if you're looking at 100 reads, so 100 read depth, they may only have 3 to 25% of those reads that are abnormal. Normally for a dominant condition, you'd expect 50% or greater, and for a recessive, so it's all different. So then you're looking, you know it's not error, because our sequencing is very good, but you're looking for 3 reads that are altered instead of 50 reads that are altered. That's a hard thing to do if you don't know what pathway to look in. And so essentially, at least for our MCAP, they're mosaic. For the MPPH, that's the one with the polydactyly, those are germline, but that's what it gave us the insight into what pathway we should look at, and I'll show you the pathway in a second. And that MPPH is a recurrent mutation, so all 11 of the patients have the same exact mutation. And so where it is over here is I just circle it there, and so this is the entire PIK3ATK pathway, and now we've actually had some other patients that we studied that have mutations in related genes in this pathway. So we've opened up a whole new pathway in the way this works. And most of them are mosaic. And so we've had to do lots of, this is a hard thing to do because looking at this level of mosaicism is not easy. Sanger sequencing, if you don't have mosaicism greater than 20%, you're never going to see it, and even still that's hard. So we've had to sequence different tissues. It has to be deep sequencing, so we have set it up to run on a MySeq. But it's not a thing, just thinking of bringing that into clinical care is still a ways away. It's a hard thing to do. So it's still being done on a research basis. But I think it really gives a great example of how the power of this technology, and even though it's a genetic disease, it's not the classic way we think about things. And so overall then, what we look at is that we solve 1 disorder per week. That's not necessarily a novel gene per week, that's 1 disorder solved per week. And they break down like this. So we studied 132 so far, and 77 have been solved. And actually those are last quarter's numbers, it's actually 88 now, but anyway. So novel genes are about half. New phenotypes, so basically that means that it may be a disease gene. For example, Notch2 with haploinsufficiency causes something called Alagille syndrome. And we found a gain of function in Notch2, which causes Head-Juccini syndrome. So it's a whole different mechanism with the same disease gene. We've seen a lot of really atypical presentations of known disease genes, and often that's because there might be only 3 people in the whole world who ever had it and were described, and it's just not on our radar to think of it in terms of a diagnosis or get access to its testing. And then for about 24, we found mutations in known genes. So these are the ones that we have published or are about to be published so far. I think there's maybe 13 or 15 or so. Our goal is to have 50 publications in 2 years from Forge, and then it'll just continue to spill out after that, but 50 is the goal I'm after. As you can see from the very, very top, the top two are those that were mutations in known disease genes. The next is the group where they are different mechanisms in known genes. And then everything in that third section is the gene was unknown, but the phenotype had been described. And then at the bottom are these new syndromes, and they are still coming up, where you actually can sort of say, okay, that's a recognizable syndrome, and then we identify a novel disease gene shortly thereafter. Really, because you only need 2 patients of one of those to find it these days. So I just want to talk about some lessons learned in our last sort of 10 minutes here, and see sort of overarching what happens. And so the first thing is it's all about the phenotype. If these patients aren't well characterized and well worked up, you haven't got anything. So next generation sequencing means next generation phenotyping. And so as we go forward in Forge, we have Mike Brudno, who's a lovely picture there, and I'm pretty sure he's much older than that now. That's the one he sent me. Looking very young, Mike Brudno, has set up this phenotyping tool based on standard ontology that you can use across the world that's accepted, so that everybody's using the exact same terminus. It's a web interface, and you click boxes. So you can say microcephaly, click, 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 click. And so this will really help us when we go to sequence patients without a family history or patients from really small families, find the other patient in the world with the same thing. In his web interface, you can put in growth curves, you can put in pictures, you can do all kinds of things. So if Forge continues after this, everybody has to fill these out. We've also learned that what happens when you try to saturate the human phenotype. So how many genes are really going to cause rare disease and how many no. And so we've solved about 60% so far. And I think what's going to happen is this hunk underneath here is probably going to collapse into a smaller number. We see that because of our 30% are sort of not novel. And for the 56 other diseases that we haven't solved, some of them are just really hard. So in about 14, these were all that had disorders that had a very nice match location. There is no coding variant that causes this. So we know this is non-coding. This means these are going to go into whole genomes, and then we're going to have to analyze the whole genome in the non-coding. That's a lot of work. The other place we've failed is where we've sequenced families with recurrent things like cleft palate. And cleft palate may really actually be multifactorial and require more than one hit, and we can't find those, so who knows. And then the other situation we have, these are still under analysis, is that there's too many variants. And for those, this often happens in dominant disorders. Dominant disorders are hard to do this way if you don't have mapping information. The heterozygous variants are all over the place. And the other thing that may be happening is that if it's anything that may be in our database for another reason, we've lost it in the filter. And so we may be ruling them out, and they're actually disease causing. We've learned some interesting things when you start to hit the same pathway more than once for a disease. So that's pretty cool. And so the example here is this is a little guy with Naegger syndrome, and this is our little guy with MFDM. They both have these facial abnormalities. MFDM has the microcephaly, and Naegger has these arm limb defects as well. And so these are both genes that we identified through Forge. And MFDM works right there at the U5. It's part of this U5 complex. And Naegger is part of U2. So there they are, overlapping phenotype. And we certainly have seen MFDM patients in the Naegger cohort, Naegger individuals in the MFDM cohort, because they interact with each other. They're hitting the same pathway. And so there are overlapping features there. This is floating harbor syndrome, which we identified. Kaufman-Sayres, Forge did not identify. We had collected our cohort, and then somebody else reported it. However, they are both part of chromatin remodeling, and so they actually have some phenotypic information that's overlapping. The other disease that falls in here, called Rubinstein-Taby, is again another one where you're seeing. You sometimes have a look and you say, was that this or that? And then we have this whole cohort now of floating harbor-like patients who are not typical, and they don't have a mutation in our gene called SRCAP, but we may be revealing another insight into this pathway, and so we're going to have a look at those. The other thing that's really become apparent to us as we go forward is we are generating new genes up the wazoo, and how are they going to go into the developmental biology field and be studied so that someone understands what they actually do in biology. And so recently what has happened is that there has been, the developmental biology community of Canada has sort of got onto Forage and said, you know what, we need to be more, we need to be friends with you guys. And so I said, okay. And so essentially what they're trying to do is they formed a consortium where they have representation from every model system. Then they have their model system community. And how they would like it to work, and we're looking for a directed grant from CIHR to do this, is that when Forage finds a new gene, or our next project, which we hope will be called Care for Rare, finds a new gene, we go to the steering committee, and then we can put out a call to the best model system in the country, who has the tools in-house, the model system is the best for the particular gene, and they get a directed $50,000 grant, and they get a year to work on it, hopefully to get enough data so they can actually get a CIHR competitive grant. And there's certainly great buy-in from the community as well as CIHR that this would be something that would work, because we want to capitalize on how well Forage has done, but share it with our developmental biology community. And the other thing that's been really interesting in terms of Forage is that we're getting biological insight into common diseases. And so that's pretty exciting. So this is Head-Duchenne syndrome, which is characterized by a whole bunch of things, including poorly developed teeth and short fingers and other sorts of things. But the other thing they have is osteoporosis. And so it's pretty interesting, actually, that this Notch2 gene is now a target for osteoporosis. It's Notch2 signaling is part of that pathway, so we've identified a potential drug target. This is Weaver syndrome, which is an overgrowth syndrome where we identified the genes called EZH2. And Weaver syndrome patients have a small increase in hematological malignancy. But right where our mutation is, right beside it, is the place where it gets hit in somatic hematologic malignancies. So now we have a rare disease in a target that's been hit by cancer and sporadic cancers, which is interesting. And then lastly, this is the pathology of a child who died of intractable seizures that was studied in Forage. All of this brown stuff is tau, which is deposited you see in Alzheimer's disease. And so this whole recycling mechanism this child has, he had a recessive loss of function of something called synaptogenin, is now emerging as part of this Alzheimer's disease pathology. And so now we have a recessive syndrome for something that is a very global common disorder. Whether or not it represents a drug target, I don't know, but does it help us understand more about Alzheimer's disease? Probably yes. And the other thing that we worked on is we really want to look at expanding our understanding of the phenotype and the natural history of rare diseases. And this is just an example of a phenotype study that Sarah Nichol at my institution has taken on. And she's studied now 65 patients with floating harbor syndrome and collected all their clinical data and is reporting something that we really didn't understand like 2 years ago about floating harbor syndrome, because most people actually couldn't diagnose it, including myself, because it's a hard phenotype to pick up. And so that's been really helpful. And then I think just one of the last things I want to talk about is the impact of clinical exomes. So 24 of the 77 disorders that we studied, we made a diagnosis because the gene was already known. And so we've officially and much more cost-effectively come to a diagnosis for that 30% of patients. And so we did a clinical exome pilot as part of Forage, where we studied 25 patients with no family history, no consanguinity, but we thought it was genetic. We sequenced either just the patient or the patient and the parents. And our hit rate is about 35% to try to come to a diagnosis. And that's about what they're reporting in the US now, where they're offering clinical exomes for $9,000 at GeneDx or Baylor. So we have a very similar rate of diagnosis. But it certainly indicates that, you know, now microarray is a first-line test for a lot of conditions. And clinical exome should be a first-line test for a lot of conditions. We need to define what those conditions should be, who are those patients, and then offer them the best test. And so we want to continue to build on that. And we did an economic analysis of this as well. Not huge, but for those patients at CHIO had been diagnosed via Forage after they'd had a workup. And we added up all the diagnostic tests that they had and got the cost from CHIO. And so essentially, if it's a degenerative disorder, we spend $25,000. Now, if you ask other people, they'll say, I spent $100,000 on some kids. We don't quite spend that much where we are. But it's, you know, $25,000 and you can get an exome for $1,000. It's not hard to do the math. And then the last thing I want to talk about that we learned is that Forage is internationally doing really well. And so here are the other centers that are doing some rare disease work. And what's happened is that they don't collaborate with one another, and their clinicians don't share information, and they don't share patients. So this is the Centers for Mendelian Genomics, which is the big NHGRI-funded US group. And essentially none of their clinicians, there's no coordination going in as to what samples should be sequenced together, and there's absolutely no sharing of data out the other end. And so what I've learned is that Canada plays well in a big sandbox. And then instead of us, I recently went to England to talk about how we would share data with the big Sanger group, and the big Cambridge group, and the big Oxford group. Because I figured they must all have some place where they're all going to put it together, and we're going to share, we're going to find second mutations, we're going to validate genes. Doesn't exist. In the end, I came away where they're all going to send and share data with me, but not with each other. It's like, what is that? I just wanted an easy place to go and query their database and come back, because I didn't want to have to set it up. Now I got to set it up, which is a pain. But hopefully this will help me. And this is the International Rare Disease Research Consortium, of which the most exciting part for Canada is that Paul Lasco, who is the current scientific director of the Institute of Genetics at CIHR, is the incoming president or chair of the executive committee, which is the committee of all 28 countries who fund rare disease research and make decisions about how rare disease research money should be spent. So that's a very cool role for Canada. I'm the chair of the diagnostics committee, which reports to the executive committee, and we tell them how we think that they should spend their money. So there are hundreds of millions of dollars coming in the next 10 years for rare disease research. How do we think it needs to be spent, and how can we leverage our stuff for Canada? And then, I swear this is the last thing, the future of medical genetics is that I think it's the best example of personalized health there is going. This is something we've been doing all along. We use genomic information to tailor the care of our patients, to impact all of their reproductive counseling, their natural history, prevent complications, and yes, we can do that for some things, and sometimes we even tailor therapy. So what I think we need to do now is to look at how to integrate genomic sequencing into the rare disease clinic. How are we going to do that? How are we going to deliver it across the country so everybody has access? How are we going to develop these databases so that we can interpret? We can't set this up independent of each other. And we need to train the next generation of these healthcare professionals to understand how to do this. And the other thing as a genetics community we need to do, and something Ignite has done so well, is we need to think more about translational rare disease treatment research and do that in a much more coordinated manner across the country. And so with that, I will close and thank all of our funders. Thank you.
Thank you, Kym. And
32 minutes, Joanne.
We'll take questions from the audience now, and for those who have to leave at 1, if you've got somewhere to run, because that was your plan, please run. But Kym is more than happy to stick around for another half an hour or so that we can ask questions in a more informal environment from 1 to 1:30.
As long as there's soup left. There's no soup left, I'm out of here.
Yeah. So please fire away.
Steve, given how rapidly this field continues to accelerate, you made a comment about training the next wave of talent. Do you think major surgery is needed in the medical schools to enable the enhanced level of training that's going to be required to interface with the technologies that you have access to?
Eventually, I think we should probably initially, though, focus it in the medical genetics community or those closely allied professions that look after patients with rare diseases. And so there's been a lot of talk about integrating this into the Canadian College of Medical Genetics and the Royal College training programs. If our next application is funded, we will develop that curriculum and implement it into both of those trains. So I think that would sort of be the first way to show how this is done, and then it would hopefully start to follow from there.
Andrew.
Can you give examples where exomes have failed to reveal a gene, but genomes have?
No, not yet. And so what happened is we only had a very small number of whole genomes available through Forge. We gave 4 to you, so maybe that would be my example. And the other one we're doing is actually, no pressure, is Aicardi syndrome, which again, we're sequencing affected brain from individuals who have Aicardi syndrome because we think, again, it's a somatic mutation, and it's much easier if you could actually sequence the affected tissue and compare it to blood and compare it to parents to try to find those. So that's where our whole genome resources have gone. In the next project, we have much more, a larger number of whole genomes available. Sarah?
I don't need to kind of step forward to the next project, but You know, I think one of the reasons why Forge has been so successful, like you were saying, is that genetics people got together, you know, we often get referred to pediatric patients with strange things. How's that going to work for the adult population when some of the adults aren't necessarily referred to clinical genetics?
Oh, that's a good question, Sarah. Yeah, because I think that, I mean, we could reach out to our adult colleagues in certain subspecialties, particularly urology, I guess. But I think Forge has worked so well because of the sociology of the CCMG and our connection and our 2 degrees of separation from one another in terms of you trained her, that person and that person's your sister kind of thing. So I think we need to keep it primarily within the CCMG, though we have our other groups that are, well, you know, our good friends in ophthalmology and all that sort of thing. But if we went too broad, I think we will absolutely lose control. So we always tinker on the balance of losing control, quite honestly.
David?
Just thinking ahead to needs, when you say retain patient information and exome reads, what format are you proposing that we retain those? The sort of final distilled version or the individual reads?
No, so we don't keep, we can go back, we keep BAM files, so we can go back and recreate the original reads if we so thought that was a good idea. But no, we keep the distilled version, and then we'll keep the HPO, like the human phenotype ontology version of the patient's phenotype, and those will be our primary data sets.
And is the HPO available just for us to browse?
It is. I can actually give you guys the website later, and you can go in and play with it. Because it's something we want to launch more wide scale, and it was something that the clinical group from the Forge Steering Committee all played with, and if there are suggestions, Mike would be happy to implement them into the interface. And also if we need changes in the terminology, though HPO's got 8,000 terms in it, but you know, the new dysmorphology terms that are accepted, etc., that Peter Robinson, who designed it, is very willing to do that for us.
Pack.
What's the difference between the analysis of the neurodegenerative diseases versus the other ones? One is more expensive.
Oh, I think because clinicians, when there's a child who is obviously losing skills and is degenerating in front of you, that there's a higher likelihood that we will investigate more aggressively. Versus a child who's presenting, say, with a congenital heart disease that was fixed or resolved on its own and developmental delay.
But sequence-wise, it's going to be the same thing.
Sequence-wise, exactly the same thing. So that $25,000 you're talking about is the other non-exome testing, yeah. So you're more likely to look at that child and think, oh, well, maybe it could be an atypical presentation of this, I better try this gene and this gene. Because you're sitting across the table from parents who want to know, who don't know that what their kid has, and how can you say we can't treat it if you don't know what it is? Right.
So shouldn't there be a hand-in-hand thing? Because you're talking about having the ontology terms, and this is exactly the same thing that happened in molecular biology, where they have to have an ontology for the proteome projects and all the other projects so that people could communicate with each other and databases can be accessed in some kind of meaningful way. But when you're talking about phenotyping your patients, don't you also, and this is, I think you alluded to, with the textbook and how to phenotype certain diseases, that you really have to have hand-in-hand the education of how to phenotype patients and how to do that cost effectively, plus the phenotype terms in your ontology.
I think that's a good point, but I think probably most physicians within the discipline, for sure genetics and ophthalmology would be the same, neurology, all of their terms will be in that.
But not from a cost-effective point of view.
Oh, so basically we've piloted this. It takes 10 minutes to put your phenotypic data in. So it's pretty, I mean, it's not hard. You can type your search term in and it just pulls down and you click.
Again, I meant like in your neurodegenerative diseases, if there are a set of 4 or 5 things, right? So if you have a diagnostic algorithm that you're going through and you're saying, okay, well, from the point of view of getting the most important phenotypic data to match it to a disease that's already in the database, you only need to do these 3 things. So that's where they're hand in hand. So there's not communication on both sides. You're still going to spend just as much money before you can get to the phenotype to understand what the disease is. That's the point I was trying to make.
Yeah,
so they inform each other. So I think there's a secondary thing that you're going to get from data mining your phenotype ontology, is that you're going to be able to make it more efficient for people to find the 4 or 5
so what
key diagnostic
features
to hit the disease when they search for it.
Andrew.
So if I could ask another question, Kym, in the paper that came out on clinical utility, I think it was exomes, I'm sure we're thinking of the same group, to address some of the issues about, for example, BRCA findings and young children. They specifically did not sequence, certain, sort of genes primarily to avoid that issue.
Oh, yeah, great. Is that the NHGRI group with the adults?
Indiana group. The paper came out about a month ago.
Yeah, I think what we would do if we're going to go forward in this is we gather the Canadian representation from across the country and decide if that's what we want to do. Are we going to totally mask those? Which we could do as a data filter, but obviously we can't primarily not generate them because the commercially available kit is the commercially available kit. And that's what we validate and that's what we like to use. Yeah, but I think that's something that we really hope that through the CCMG to sort of build a consensus statement and a position statement on what we think, how we do this, and what genes we report and which ones we don't, and how you opt in and out of these things. Great gels question.
More? All right.
I was going to see
Great.
one more question. Put my cancer hat on.
Okay.
And so you did allude to interaction with the BC Cancer Agency.
Mm-hmm.
So are you interacting with Terry Fox and some of the other agencies to coordinate the cancer genomics with some of the rare disease? Because when you talked about the hematopoietic disorders, there was this really nice correlation with the EZH2 and the actual, I think was it pre-BAL, something like that?
Yeah, so yes, I think we do. It hasn't come across that many times, but they're
And I would
definitely,
think
those are
in
the
your-
databases that we query.
In your PR3 kinase stuff, that's a classic
Yes.
pathway that's
Yep.
involved in
Yep.
TLL.
Yeah, so then
And
we go to the
so
database. We
They
we
already
ask that
have
question.
drugs and, should be
Yeah.
communicating because the translation efforts are already really
It is, it's
down the
absolutely
road.
true. They have drugs up the wazoo for that pathway,
Yeah.
all those inhibitors. And so the mice are being ordered and all, yeah. So that's the other thing, right? How do we then do high throughput translation for all of these things that have inhibitors that are sitting on shelves of pharma that have been created for another purpose and quickly have a look at them in some systematic way?
Joanne.
How do you get your moving exomes into a clinical diagnostic, available clinical diagnostic test? What do you think would be needed to get to that level? Because that's,
Yeah.
right now it's research with the volume, who's going to interpret the data, how do you see this working?
Well, I can only speak for Ontario because that's the one I know the best. And so we've had conversations with the Ministry of Health, the Health Services branch that deals with our out-of-country funding. So it's a different mechanism that you have here where you're giving us some lump sum of money. But ours goes centrally. And so they have spoken to us about, they're aware of this technology, they're aware it's emerging. And unfortunately, how it works, well, fortunately or unfortunately, how it works in Ontario is that the more, their feedback to us is that if it's medically indicated, we will provide it. So the first thing we need to do is have the CCMG issue a position statement to say which rare disease patients is this medically indicated for. We have to create that data to create that position statement, but that's number one. The second thing is it's available in the US in a couple of places now, and so we are now beginning to ask the Ministry of Health to pay for this. They have a process by which they have to review it, and it usually takes about 9 months to a year. But I've pinged them multiple times now with patients that I think need a clinical exome. And a success rate of 30% for a diagnosis is pretty reasonable. And if I order a limb-girdle muscular dystrophy panel, that's 12,000 euros. So $9,000 for a clinical exome is actually cheaper. And they're very aware that they want to repatriate. So once they get an out-of-country budget line for exomes, that's when they look at, oh, can we do this cheaper if we do it back in the province? And that's how we drive that process. In the interim, we would need to make sure that there are diagnostic labs within Ontario ready to do this. Usually, unfortunately, one must find their own infrastructure. That's how microarray came in for most places, probably, right? And so we'll need to find some place where they're comfortable. There will at least be 1 or 2 labs in Ontario that will be ready to do this at that time.
And if they could send would there be interpretation on site? Because that's an expertise.
Interpretation on site, yeah.
Because you wouldn't be able to send just the results to the physicians.
Yeah. And I think over the next couple of years, those interface tools will be made much better than they currently are.
All right, so I think that wraps up, and I'd like to thank Kym again.
Thank you.
For the audience for coming.